السلام عليكم طلاب مرحلة ثالثة. كل عام وانتم بألف خير وصحة وسلامة. أنا دكتور حيدر، إن شاء الله أكمل وياكم هذا الأسبوع واللي أسبوع بعده. ف be 3 subjects. The first, our subject is pentose phosphate pathway. Okay? Pentose phosphate pathway. In the beginning, you could know the references. We've got 2 references اللي هو Biochemistry 5th edition. هذا يسمى Strial or Liberty Strial، مشهور famous name Strial. Second, Harvard's Illustrated Biochemistry اللي هو التكست بوك مالتكم. Okay, pentose phosphate pathway. What's the biomedical importance, or why do we need study the pentose phosphate pathway? Pentose phosphate pathway, let us start with the meaning or the names of the pentose phosphate pathway. Pentose phosphate pathway has several names, more than one name. First, pentose phosphate pathway, and hexose monophosphate shunt يعني الوصلة. Or the third name is phosphogluconate pathway. Now we've got 3 names for this process: pentose phosphate pathway, hexose monophosphate shunt, or phosphogluconate pathway. Any name from these means this process, which is a famous name, a pentose phosphate pathway. Okay. What's the relationship between the pentose phosphate pathway and hydrolysis of the glucose? In the fact, pentose phosphate pathway is an alternative route for the metabolism of the glucose. Therefore, it's clear a relationship between the hydrolysis of the glucose and pentose phosphate pathway, because it is one of the route for the metabolism of the glucose. Okay, have a look for this reaction. We've got glucose in the glycolysis or Krebs cycle, oxidative phosphorylation. The result is ATP, adenosine triphosphate, unit of the energy. Now, this is unit of energy. This is the result from the hydrolysis of the glucose. The result is ATP, adenosine triphosphate. That means we need oxygen here, oxygen to give this energy unit. We need to convert some of the important compound to oxygen, then the result or the production is ATP. Okay, this is for the glucose. Now, in the pentose phosphate pathway, phosphate pathway doesn't generate ATP, but has 2 major functions. Now, we're going to explain what is the major function of the pentose phosphate pathway. Despite there is a relationship, very clear relationship between the hydrolysis of the glucose and pentose phosphate pathway. What is the major function for this process? First, synthesis of the NADPH. Okay, and NADPH is the main function for the NADPH is working as a reduced agent. It's working as a reduced agent. Okay, yes, the formation of the NADPH or nicotinamide adenine dinucleotide phosphate, the abbreviated NADP plus, or in old notation TBN, is one cofactor used anabolic reactions such as the Calvin cycle and lipid and nucleic acid synthesis, which require an NADPH as a reducing agent. It is used by all forms of the cellular life. It's very important, you have to know NADPH is the reduced form of NADP plus. We're going to explain the more details about the function of the NADPH. Okay. Now, nicotinamide adenine dinucleotide phosphate, nicotine, this is nicotinamide, adenine, this is this adenine unit, nicotinamide adenine dinucleotide, this one molecule and this one, dinucleotide phosphate. Now, nicotinamide adenine dinucleotide, this one and this one phosphate. Okay. The second important function for the pentose phosphate pathway is formation or synthesis of ribose for the nucleotide and the nucleic acid formation. A nucleotide or a nucleic acid formation. This is the structure of the ribose 5-phosphate. Structure ribose 5-phosphate. The phosphate group in the carbon number 5. Ribose consists of 5 carbon atom 1, 2, 3, 4, 5. Ribose, and the phosphate group in the carbon number 5. This is very important to synthesis of the nucleotide, like addition to RNA, DNA. Therefore, it is very important unit or essential unit to synthesize all these biomolecules like DNA, RNA, or nucleotides. Okay. Now, the formation of the NADPH, it is used for biosynthesis of fatty acid and steroids. Now, one of the important for the NADPH, one of the important of the NADPH function as a factor or factor to biosynthesis of the fatty acid and steroids. Steroids, the essential unit is cholesterol. Therefore, some of the high potential electrons or fuel molecules يعني الجزيئات المليئة بالطاقة الإلكترونية عالية بطاقة إلكترونية عالية must be conserved for the biosynthetic purposes like reductive biosynthesis rather than transfer oxygen to generate ATP. I said that clearly in the past page. Here the compound will not be converted to oxygen to form ATP like a glucose. We need molecules high potential electrons to synthesis like lipids, fatty acid, or steroids. This is one of the examples. One of the examples here. Now, in the pentose phosphate pathway, NADPH is generated when a glucose 6-phosphate is oxidized to ribose 5-phosphate. Now, NADPH is formed after oxidation of the glucose 6-phosphate to ribose 5-phosphate. Okay? This is very clear. Have a look for this reaction, general reaction to synthesis of the ribose 5-phosphate and molecules of an ADPH. Glucose 6-phosphate, as we said, when glucose 6-phosphate is oxidized to ribose, this is glucose 6-phosphate, in the presence of oxidized form of the NADPH, or just NADP plus, this is the oxidized form, plus 1 molecule of water. The result is ribose 5-phosphate and 2 molecules of NADPH in addition to carbon dioxide and 2 moles of the hydrogen plus or proton. Therefore, after oxidation of the glucose 6-phosphate, the result is 2 molecules, that's it. 1 molecule of glucose 6-phosphate will be converted 2 molecules of the reduced form of the nicotinamide adenine dinucleotide phosphates. Okay. Now, this reaction, general reaction, consists of 2 phases. 2 phases. Or the sequence of the reactions of the pentose phosphate pathway may be divided into 2 phases: oxidative, non-reversible phase and non-oxidative reversible phase. Okay, let us start with the first phase. In the first phase, glucose 6-phosphate is going to form or 2 steps or 2 processes will be seen in the first phase. The first one, dehydrogenation, and the second one, decarboxylation, to yield a pentose or ribulose 5-phosphate. Now, the first phase is the substrate is converted to ribulose 5-phosphate after 2 steps, dehydrogenation and decarboxylation. Now, have a look for this diagram. We've got 3 molecules of glucose 6-phosphate, 3 molecules of glucose 6-phosphate. Now, in the presence of glucose 6-phosphate dehydrogenase, this is our enzyme to convert glucose 6-phosphate to ribulose 5-phosphate. Here we've got NADP plus and 1 molecule of water in oxidized form for the NADPH. The result, in the presence of this dehydrogenase, in the presence of this enzyme, 6-phosphogluconate and 1 molecule of NADPH reduced form, in addition to reduce or consume 1 molecule of water. Of course, we've got coenzyme, magnesium and calcium. This step, it is like this step and like this step. Okay, anyway, the result is 6-phosphogluconate. Then in the presence of the 6-phosphogluconate dehydrogenase, another molecule of NADPH will be formed. 1 molecule of the oxidized form, NADP plus, will be converted to NADPH, 1 molecule of NADPH plus H plus. In addition to removing 1 molecule of carbon dioxide. But the result is converted 6-phosphogluconate to ribulose 5-phosphate. Same ribulose 5-phosphate. Okay? Now, 2 enzymes. The substrate glucose 6-phosphate, this is glucose 6-phosphate dehydrogenase. Okay? The second enzyme, 6-phosphogluconate, this is 6-phosphogluconate dehydrogenase. Okay? two molecules of NADP plus were used, and the result, two molecules of NADPH reduced form, and there is removing one molecule of carbon dioxide and consuming one molecule of water, and the result is ribulose 5-phosphate. This diagram is considered the first phase of pentose phosphate pathway. This is the dehydrogenation. We've got two enzyme dehydrogenation, okay, and decarboxylation, removing one molecule of carbon dioxide. Okay, this is the mechanism of the ribulose 5-phosphate synthesis in the pentose phosphate pathway. Have a look for this glucose 6-phosphate D, that's mean hydroxyl group in the carbon in the right hydroxyl group, it was here. Beta for the active group, aldehyde group, beta not alpha. 6-phosphate, 6 carbon, and the phosphate group in the carbon number 6. In the presence of the glucose 6-phosphate dehydrogenase and coenzyme, magnesium +2, calcium +2, and oxidized form of NADP, the result is 6-phosphogluconolactone, ketone, and one molecule of NADPH. Okay, as intermediate compounds. First, one molecule as intermediate compound results from the conversion of the glucose 6-phosphate to ribulose 5-phosphate. Okay, the second step, 6-phosphogluconolactone, this compound, in the presence of gluconolactone hydrolase, that means it will be added one molecule of water for this compound in the presence of the magnesium, manganese, and calcium +2 as a coenzyme for this enzyme, and the result is 6-phosphogluconate. Gluconate, we've got carboxyl group in the carbon number 1 and phosphate group in the carbon number 6. Then the next step is conversion of 6-phosphogluconate in the presence of dehydrogenase, another molecule of NADP plus oxidized form, to form the second molecule of the NADPH plus H plus, and the result 3-keto 6-phosphogluconate. 3-keto 6-phosphogluconate. Gluconic acid is carboxyl, ketone, and phosphate group. The final step to reach our destination is removing one molecule of carbon dioxide. That means decarboxylation, and the result is ribulose 5-phosphate. Therefore, dehydrogenase, 2 times for the dehydrogenase, 2 molecules of NADPH will be formed, this one, and this is very important. Therefore, NADPH, it will be formed during the conversion of the glucose 6-phosphate to ribulose 5-phosphate. 2-step dehydration, 1, 2, and 1-step decarboxylation. Okay. Now we've got a simple idea to form or to resolve the molecule of NADPH. Now, what's the biofunction of the NADPH? Let us see the biofunction or the importance of the NADPH molecule. As I said in the beginning, NADPH molecules is used as a reduced form. Therefore, NADPH is used to keep the cellular and mitochondrial glutathione in the reduced form through the action of glutathione reductase. Now, the first important function for this molecule, nicotine or NADPH, nicotinamide adenine nucleotide diphosphate, to keep the glutathione in the reduced form. Why? Let us see the reaction. Have a look for this reaction. We've got the glutathione, but in the oxidized form. What does it mean, oxidized form? That means we've got two sulfur, the bond between two molecules. This one bonded with this one by the sulfur bond, and this is the oxidized glutathione, or disulfide form. Disulfide form, GSSG. Now, three terms: oxidized glutathione, or GSSG, or disulfide form. In the presence of NADPH plus H plus, glutathione reductase enzyme, very important, this step, this compound, oxidized form, will be converted to reduced form, or reduced glutathione, GSH, from GSSG to GSH, or sulfhydryl form, and NADP plus. Okay, NADP plus. Now, this is the first function for the NADPH, to keep the glutathione in the reduced form. And why does it that? Or what's the advantage from the keeping the glutathione in the reduced form? We're going to see this, the answer of this question. Okay, reduced form of glutathione plays a several important role. Now, because of there is enough amount of the NADPH, that means all the oxidized form of the NADPH will be converted to reduced form. Now, what's the advantage, or what's the biofunction, or what's the function for the reduced form of the glutathione? More than one function. I'm going to explain three of them. It is essential for the maintaining the normal structure of the red cells, and for keeping the hemoglobin in the ferrous state. Okay, now to protect the red cell, or RBC, from the hemolytic anemia, by keeping the hemoglobin in the ferrous state. What does it mean, ferrous state? The iron will be in the Fe plus 2 form, will be converted the Fe plus 3 to Fe plus 2 form. That means ferrous state. By this way, the structure of red cell will be in the normal state, will be outside of the area of hemolytic anemia or hydrolysis. It serves as, in addition to that, it serves as sulfhydryl or sulfhydryl buffer that maintains the cysteine residue of hemoglobin and other red cell proteins. Now, in the reduced state. Now, the second function for the reduced form of glutathione, to keep the amino acid cysteine in the, to keep the amino acid residue, or amino acid cysteine, or cysteine residue, in the reduced form. In the reduced form, that means has thiol group, GSH, without S bond S, because of in the presence of GSH can be used to reduce the protein disulfide back to the thiol form. Therefore, any oxidized form for the cysteine as a cysteine, S bond S, will be converted in the presence of GSH to cysteine residue, like the active group or side group will be thiol group, GSH. Okay, this is the second function for the reduced form of the glutathione. First one, to keep the RBC from the hemolytic anemia by keeping the hemoglobin in the ferrous state. that means iron Fe+2 instead of the Fe+3. In addition to that, to keep the cysteine residue in the reduced form, cysteine instead of cysteine. That means the side group is thiol group instead of S bond S. Okay, have a look for this function. For this reaction will keep the cysteine in the residue in the reduced form. Protein has S bond S protein. That means this is the oxidized form in the presence of two molecules of reduced form of glutathione. This protein will be converted, this residue or this reduced glutathione will be converted, this residue, to cysteine like SH or thiol group, and GSSG will be go back to form two molecule of reduced form. Okay, now because have a look for this reaction. This is to go back to form a reduced form of glutathione. Okay, in addition to that, we can give you the third function for the reduced form of glutathione, play as detoxification by reacting with hydrogen peroxide and organic peroxides. Now, it will remove any peroxides compound. This reduced form will remove this toxic compound by react with them on, for example, hydrogen peroxide will be converted to two molecules of water in the presence of the reduced form of the glutathione. Therefore, will be removed the toxic compound. Have a look for this diagram. This is rule of glutathione to remove the hydrogen peroxide. Here we've got pentose phosphate pathway will convert the oxidized form to give reduced form of NADPH, and NADPH will transfer or convert the oxidized form of the glutathione in the presence of glutathione reductase to reduced form. This is very important. It's coming, of course, according to for this diagram, reduced form of the glutathione is coming because of NADPH. NADPH will convert an oxidized to reduced form. Now, reduced form will remove any peroxide compounds or peroxide, like hydrogen peroxide. This reduced form reacts with the hydrogen peroxide to form two molecules of water, and the reduced form will go back to oxidize, and because of the presence of the reduced agent, convert another molecule of oxidized glutathione to reduced glutathione, and the process will be continued until all the molecules of hydrogen peroxide will be removed. Now this is the third function. This is the third function for the reduced form of the glutathione, and its relationship with the reduced reagent. This is reduced reagent or NADPH. Now we can make a relationship between the peroxide, hydrogen peroxide, and NADPH. Because of the presence of this compound, the oxidized form will be converted to reduced form, and this agent will convert the toxic compound hydrogen peroxide to stable compound, two molecule of water. Okay. The conclusion from these three functions, we can say, therefore, cell with a lower level of reduced glutathione are more susceptible to hemolysis, more susceptible for or more exposure for the hemolysis. Any deficiency in the reduced glutathione, that means there is a lot of toxic like peroxide, and this will make problem. This will make problem because will hydrolyze the RBC because there isn't enough amount of reduced form of glutathione. That means the hemoglobin, some of the hemoglobin will be converted to iron in the hemoglobin will be converted to Fe+3. Therefore, will make hydrolysis for the hemoglobin. Then the age of the RBC will be less than the normal, and the spleen will be removed the RBC before 90 days, which is the normal, approximately the normal age for the RBC, and therefore will make deficiency in the amount of the RBC and deficiency in the oxygen. Let me give you example for this example for some drugs like Permaquine. Drugs like Permaquine years ago it was used to treat the malaria, but this drug or medicine distorted the surface of the blood cell in the absence of the reduced glutathione, which makes them more liable or exposure to the destruction and removal by the spleen, as I said, because these drugs increase the rate of formation of the toxic of peroxides, which are normally reacted with the reduced form of the glutathione. Okay, now this medicine or drug will make the age of the RBC less than the normal age. Why? Because this will be increase the rate of formation of the toxic of the peroxide, and the peroxide according to this diagram, a lot of molecules of the hydrogen peroxide or any organic peroxide will reduce a lot of molecules of the reduced form of glutathione. That means we haven't enough amount of this compound, and the RBC will be more susceptible for the hemolytic anemia, more susceptible because the cell of the RBC will be hydrolyzed by this compound. Okay, this is very simple, drug for the importance of the reduced form of the glutathione. These drugs will make a lot of toxic peroxides, and by this diagram will reduce the amount of glutathione, reduced form. Therefore, absolutely there is hemolytic anemia, or some of the protein will still in the oxidized form for the cysteine residue. It will be S bond S instead of thiol group, and the function this will affect on the function of the protein. Okay, the second phase or in the second phase, ribulose 5-phosphate is converted back to the glucose 6-phosphate, giving ribose 5-phosphate as intermediate product by series of reactions involving mainly two enzymes, transketolase and transaldolase. Okay, in the first, okay, let us see this diagram. Let us go back a little bit. In the first phase, in this first phase, there is conversion of the glucose 6-phosphate to ribulose 5-phosphate. During synthesis of the ribulose 5-phosphate, there is formation to 2 molecules of an ADPH reducing agent, and this is very important to keep the glutathione in the reduced form. And we have seen now the function of the reduced form of the glutathione. For example, 2 functions in the RBC, in addition to remove the toxic compounds like hydrogen peroxide. Here, the second phase, ribulose 5-phosphate is converted back to glucose 6-phosphate. Now, ribulose 5-phosphate is converted back to glucose 6-phosphate. Okay, to glucose 6-phosphate. Now, absolutely, there is the main enzyme are used in this mechanism is transketolase and transaldolase. Let us see some steps here. 3 molecules, same 3 molecules, ribulose 5-phosphate, ribulose 5. In the presence of epimerase enzyme is converted to xylulose 5-phosphate and ribose 5-phosphate and xylulose 5-phosphate. Here, ketoisomerase enzyme. This step, same enzyme, epimerase, that means the result was xylulose 5-phosphate. But ribulose 5-phosphate in the presence of ketoisomerase, the result is ribose 5-phosphate. Here, the important things, because ribose 5-phosphate is, as I said, one of the results from the pentose phosphate pathway, and this molecule is very, very important to synthesis of the nucleotide, RNA, and DNA, and you have what's the function of each one, RNA or DNA. For example, DNA as a genetic to transfer the genetic information to ribosome to synthesize new protein. This is outside scope of our lecture. Have a look for then in the presence of transketolase enzyme, this compound will be converted to dehydrogenase 7-phosphate or glyceraldehyde 3-phosphate. But here it's very important to mention the function of the vitamin B1, thiamine P2, in addition to the coenzyme Mg+2. Now here, in the synthesis of the ribose 5-phosphate, B1 is very important. B1 is very important. So let us continue. Transaldolase, the second enzyme, transaldolase, will convert this compound to fructose 6-phosphate, erythrose 4-phosphate. Then, in the presence of the phosphohexose isomerase, this compound, fructose 6-phosphate, will go back to glucose 6-phosphate. In the other side, in the presence of transketolase, more than one step, we've got half mole of glucose 6-phosphate, more than one step and more than one enzyme. The important things here in the second phase, synthesis of the ribose 5-phosphate as an intermediate compound, and its function very important to synthesis of RNA, DNA, and the nucleotide. Okay, in the first phase there is production for the NADPH. In the second phase there is production for the ribose 5-phosphate, and this is the second important function for the pentose phosphate pathway. Okay, this is second phase of pentose phosphate pathway, this diagram, and the main enzymes transketolase and transaldolase, and have a look for the B1, vitamin B1. يعني B1 مهم جدا وراح يكون كميديسين موجود بصيدليتك. Okay, now let us see the figure 8 rule of the thiamine pyrophosphate B1 in the pentose phosphate pathway. Here, ribose 5-phosphate or xylulose 5-phosphate in the presence of transketolase and B1 in addition to magnesium plus 2 as a coenzyme for the transketolase. The result is glyceraldehyde 3-phosphate, glyceraldehyde 3 carbon atoms, phosphate in the carbon number 3, and dehydrogenase 7-phosphate consists of the more than 6 carbon atom. Two compounds in the presence of transalderase, the result fructose 6-phosphate and erythrose 4-phosphate. Okay, so this compound is converted to glyceraldehyde 3-phosphate, erythrose 4-phosphate to glyceraldehyde 3-phosphate in the presence of transketolase and vitamin B1 and magnesium plus 2 as a coenzyme. Also, this compound xylulose 5-phosphate will be converted to fructose 6-phosphate. Now, two steps to convert the ribose 5-phosphate to fructose 6-phosphate. There is 2 steps including the vitamin B1. Therefore, it is very important vitamin in the pentose phosphate pathway, especially where in the second phase to synthesis ribose 5-phosphate. Okay, I guess enough to this page. Hopefully it was very interesting, or it's interesting lecture. Any question, don't hesitate. I'll give you 10 minutes more to answer your question. Okay, brilliant. Now finish everything, and thank you so much for your listening, and goodbye.